So as we move on in these 30,000 foot view videos, we're constantly going to refer back to this slide from our very first video, and we're going to make sure that we see the totality of this metabolism picture, but then zoom in and say, well, what are we understanding a little bit more? So in this video, we're going to have a 30,000 foot view to understand the digestion of fuels. And really where that was in this top part here. When we have fuels that we consume, proteins, carbohydrates, lipids. Again, reminding ourselves that the main purpose of protein consumption is to obtain building blocks. Carbohydrates and lipids really are obtained for fuel. But we've got these polymers or macromolecules that we need to break linkages in order to get them into their monomeric form, because only monomers enter our GI tract and enter our bloodstream. So we're going to be focused kind of on this piece and fully understanding sort of how that process works in digestion of fuels. So on this slide, I'm going to have carbohydrates and lipids together because those are our fuels. You'll see on our next slide, we're going to talk about digestion of building blocks. So differentiating between proteins and carbohydrates and lipids, fuel molecules, proteins that we consume generally to obtain their building blocks to be able to use for other purposes. Okay, so when we think about fuels, again, we're going to map on some things that we already know, sort of reminding ourselves of some other pieces. When we talk about carbohydrates, right, I want you to remember that they are 4 calories per gram. And then when we have lipids, and specifically we're going to be talking about triglycerides, those are going to be 9 calories per gram. Okay, so consuming these fuels, take a step back for a second. Just think about our digestive system. We're not going to get into a lot of the anatomy and physiology that happens in the digestive system, but I want you to think about there being sort of, several main parts to it. First part sort of is your mouth, right? And then you've got your stomach, okay? Then you've got your small intestine, and then you've got your large intestine. So here are the highlights for things that I need you to know about. The importance of sort of starting this digestion process in your mouth, right? We masticate food, we chew it up, we get it into smaller pieces. Think about some of the highlights and hallmarks of what happens in our stomach. We've got a lot of mixing that happens in our stomach, that peristalsis that begins in your stomach, very low pH in your stomach. Again, part of that purpose is to make sure that you take that hostile outside environment that you brought inside and you help to make sure that it's not too problematic. So low pH is really going to help with that acidic environment. Get out of your stomach and now into your small intestine. A lot that happens in your small intestine in terms of digestion and sort of breaking those linkages. So a lot of that happens enzymatically there. We dramatically change the pH as we go into our small intestine, a little bit north of neutral. They're a little bit more around the pH 8 range. But importantly, we talked a lot about our small intestine. That's where we have that single cell thick layer where we're going to be able to get out monomers from our GI tract. A lot of nutrient absorption is going to happen in the small intestine. Large intestine, here's the things that we're going to focus on in the large intestine. We're going to talk about the importance of the gut microbiome that's there. We're going to talk about water resorption, okay? But again, one of the things that also happens related to that gut microbiome piece is anything that doesn't get fully digested, fully hydrolyzed, fully linkage broken in your small intestine, your gut bacteria are going to have a go at it. And sometimes that's a good thing when we talk about fibers or anything that's a prebiotic for your probiotics, right? But if it's something that, you can't digest, like lactose digested by lactase, then you might have a situation where that bacteria trying to break down that lactose milk sugar is going to create some hardships. So just some things to sort of think about there, but that's sort of the overview of what I want you to think about in terms of your digestive tract. So let's sort of move in here. Here's some things that I want to make sure that you understand about carbohydrate digestion that we have here. Again, begins in the mouth. So begins in your mouth. We have a salivary amylase. So anything that I'm going to highlight in green, these are names of things that we've either seen already and or that I want you to know. Okay. And so what happens is we're going to start breaking those glycosidic bonds that we have in starches. And importantly, we are going to be generating glucose. Okay, now why this is important is glucose is going to signal your taste receptors. So again, this is going to taste sweet We talked about sort of that demo that you could do. Take an unsalted saltine cracker, chew it until it's a paste in your mouth, and literally keep it there for several minutes. It will start to taste sweet. And that's because you will take the starch that's in that cracker, you'll start breaking those glycosidic bonds, you'll be liberating free glucose. It's going to signal your taste receptors, and you're going to taste sweet. Now, why this is important too, sort of this early digestion, is when you have a signal for glucose in your mouth, your pancreas will actually already start to secrete insulin in preparation for that glucose entering your bloodstream. Okay, so moving through the stomach, again, not a lot is important from the digestion standpoint in terms of breaking linkages or nutrient absorption so much in the stomach. Getting to the small intestine is where we're really going to have a lot of the money pieces that happen, right? So some enzymes that we've already talked about. We talked about the enzyme lactase that breaks lactose into its monomer forms, galactose and glucose. Sucrose, right? Nature's important disaccharide there is going to be broken up into glucose and fructose, but big picture here, and we're going to see this sort of as a theme throughout our slides that we have here in this section, we need to have monomers because only monomers are going to exit your GI tract. So these, again, are the monomers. These are the only things that are going to exit your GI tract and be able to go into your bloodstream. Okay, so that's what we have, just in a simple way to think about digestion of carbohydrates. Let's come on over here and think about what we need to have happen with lipids. So again, lipids are unique as a class of biological molecules, right? They're very hydrophobic. So two sort of lipids that we're going to focus on here and talk about is we're going to talk about triglycerides, and we're going to talk about cholesterol, two important dietary lipids that you sort of take in. So we'll talk more about lipid digestion next semester, but I want you to know that lipases are responsible for taking that triglyceride and snipping off at least one of the fatty acids. We cannot move triglycerides out of our GI tract, but as long as you snip off at least one fatty acid, our picture here shows that you've snipped off two to generate a monoglyceride. You can have diglycerides exit your GI tract, but you cannot have triglycerides exit your GI tract. So again, we're going to have these guys be sort of monomerized, if you will. We're not breaking a polymer linkage, but we are breaking that ester linkage that we have, right? It's not really shown here with a functional group, but remember this is going to be here an ester linkage that we have that hooks those fatty acids onto our glycerol backbone. So we need to have monomers in order to get out of our GI tract. All right, so here again are the monomers. We're going to talk about cholesterol in just a minute, but one of the important things that sort of happens, cholesterol is very water insoluble, but emulsification with bile salts, again, that's why those bile salts, they're created from cholesterol, but that's your sort of natural soap that's in your GI tract, secreted from your gallbladder, produced in your liver, but that's going to help sort of emulsify and sort of get cholesterol into your GI tract. Okay, lots happening again in the small intestine. So we're going to have the complexing of anything that is hydrophobic, like triglycerides or anything that's going to be lipid derived. We often are going to complex it with protein in order to create these lipoproteins that are going to be able to circulate in the bloodstream. So highlighting, this is a common theme for things that we see is we generate these lipoproteins, and they are going to be able to circulate in the bloodstream, okay? Talking about what happens with cholesterol, right? We've got fatty acids, we've got that carboxylic acid head group, and just like we saw when we had a glycerol backbone, we can esterify cholesterol to fatty acids, and we can generate a cholesterol ester. Okay, so this is how we're going to see cholesterols. Oftentimes these will be sort of packaged with some of our lipoprotein particles, but these are going to be able to exit out into the bloodstream and ultimately be delivered to the tissues. So two main fuel molecules, carbohydrates and lipids, we need to generate monomers to exit our GI tract and get into our bloodstream. Now, remember, we talked about once you have these sort of monomers entering your bloodstream and then going to sort of your distal tissues for metabolism, your body does not care whether they are from endogenous or exogenous sources, whether they're dietary or sort of from your own reserve preserves. Okay, so let's take a second here, and then we're going to sort of talk about what happens when we have digestion of proteins. And we're highlighting these are building blocks. Now, they can be metabolized as a fuel, okay? They can be a fuel, and again, they're going to be 4 calories per gram, but only if we have excess. Okay, so let's sort of talk about what's happening here. Now, here we do see an important piece that happens sort of in stomach. The digestion process for proteins in the stomach is going to ensure that we denature our proteins. And importantly, on this slide, we're going to highlight the differences between these two D words. To denature a protein means to unfold it. So now we've got this unfolded protein that's going to make its way into a small intestine. And here we are going to see the important serine proteases that we learned about, right? Pepsin, chymotrypsin, trypsin. These are some of the serine proteases. And I should say that chymotrypsin and trypsin are really serine proteases. Pepsin is a different enzyme. But we've got serine proteases, and they are going to facilitate the digestion of our protein by breaking that peptide or that amide linkage in amino acids that are held together there to make our protein. So here we have degradation. Okay, so here is degradation versus denaturation. Want to make sure you understand the differences between those. But once we get to this point, you can see here we have got our monomers. We've got amino acids. So again, monomers here, and that is what we need to get out of our GI tract. So monomer. Okay, only monomers are able to exit your GI tract, enter your bloodstream, and ultimately be delivered to your tissues. So just to remind you of some of the stories that we talked about, the ability of these monomers to exit has to do with their transit across that single layer or single cell layer thick portion of your small intestine. We talked about if you have an integrity in your small intestine, we call that leaky gut. If the junctions between your individual intestinal epithelial cells aren't sort of tight, we don't have what we call tight junctions, that means intact peptides or even full proteins can get out. And if you have foreign peptides or proteins that get into your bloodstream, that is going to cause an immune response. So if you have peptides or proteins that are able to exit your GI tract, they're going to elicit an immune response. And for many people who have leaky gut, autoimmune is often sort of a component to that. So purpose again of having these building blocks, having these amino acids, just one last little piece to sort of map on here. These amino acids are generally going to be used for one of two functions. We are either going to build other proteins. Remember, half of the amino acids are what we consider essential. We cannot produce them, so we need to consume them in our diet. Or we could use those amino acids and make them into amino acid derivatives. So use as an amino acid derivative. So we'll talk much more about that next semester. But again, that is an overview of thinking about this digestion process, highlighting that we need to be able to get monomers out of our GI tract. Sort of summarizing from this slide again, it's only those monomers that are going to be able to be in our bloodstream and thus circulated around to peripheral tissues.